Yeah. All right. I think one of you in the audience, where is somebody wanted to make, your friend wanted to make an announcement. He left, but do you want to make an announcement? Oh, yeah, you're here.
Oh, yeah, sorry, the only thing I wanted to say is if there are some lecturers who are around on the weekend, Mo was saying that she'd be open to having science discussions over the weekend. She's here until Monday, so feel free to try to set something up if you want to discuss science over the weekend. But in the mornings, you should try to hike.
Yeah. Yeah, I think some of us are still here. Like Marcus, you're also here. So, yeah. So if you're, other questions, we don't want the discussions to just end, with your presentations. They can keep going. So, okay. All right. So I guess this is my final discussion. So some of you wanted, to chat a little bit about phase transitions in biology. So I wanted to start with some examples and then some specific examples that are rigidity transitions that we talked about briefly in the last two lectures, but we will do a little bit more today. Oops. Sorry. Oh. Let's see this one. Okay, thank you. All right, so I wanted to start with some examples of phase transitions in biology, right? So in physics, we usually characterize phase transitions as of two types, right? First order phase transitions, where the characteristic order parameter of the system sort of has this discontinuous jump at the transition, right? If you look at it as a function of some control parameter. And then second order, or sometimes known as critical phase transition, where this is more of a continuous change, right? So in biology, people have looked at and characterized different types of phase transitions, in terms of this very nice paper from which I grabbed this, synchronization in neuronal networks, flocking or swarming, right, of insects and birds, rigidity of actin networks that we briefly talked about, rigidity percolation and zebrafish morphogenesis that we briefly talked about, and then also spreading of invasive species, and then phytoplankton traits. So these are some examples, right, where in biology we see these different types of phase transitions. And then now if you ask, okay, so in each of these systems that I gave you examples of, what is the control parameter? What is the order parameter? And whether this is a first order phase transition or a second order phase transition, like from studies that people have done so far, how do we classify this? So these are some examples, right? The same examples that I just mentioned in the previous slide. So these are the two in our examples that are basically mechanical phase transitions or rigidity phase transitions, right? Where as we change our tuning or control parameter, the mechanical, the shear modulus or a mechanical property undergoes a significant change. So for actin, it's basically the concentration or density of either actin filaments or density of crosslinks. As you change those, you will see a phase transition, and the phase transition is basically in rigidity or shear modulus. And this is the reason it's said unspecified, because these networks can be very messy and heterogeneous. And it's difficult to see whether it's a sharp jump or a continuous jump, because simulations might sometimes say something, experiments sometimes say something else. But still, most of the stuff that people looked at is still second order, I would say. And then the rigidity percolation in zebrafish embryo that I showed you an example of in last class. This is, again, the order parameter. In this case, you can think of it as tissue viscosity or the complex modulus in this case, because there is an elastic part, there is a viscous part. You can't neglect the viscous part. And it's a second order type of phase transition, right? So, yes. Yeah, but this is clusters. This is in terms of experiments, right? So in experiments, often you think of, let's say, if you think of collection of cells or a collection of in neuronal networks, neurons, specific types of cells, there is something we call the giant cluster size. So the cluster size, you will also see a big change in the size of clusters. These are finite system sizes, etc., right? And when we talk about connectivity percolation, et cetera, there we are mostly thinking of a Peary model, right, on a lattice, et cetera, right? So this is talking about cluster sizes more for simulation and experimental systems of finite sizes in this case. And you do see a big change.
For connectivity percolation.
Yeah. Yeah. Right. Right.
Then for rigidity percolation, like, I don't know what's the right correlation, like, that should be shown to diverge at the, like, how would you even define the exponent for rigidity?
So for rigidity percolation, I'm not going to talk about that, but one of the points where people study this critical point is the so-called isostatic point, right? Remember the point where we had seen, let me come to that figure, and then I will tell you where people usually study what's going on in rigidity percolation, right? But you are right that, at some point you will reach connectivity percolation, and then you will see, like, a growth, like, a rapid change in the cluster size. But so the main thing is tissue viscosity, but cluster size, the reason they mention this in their chart, so this is taken from this paper, is because in the experimental paper that they're using to sort of, classify the system, the experimentalists also looked at a distribution of cluster sizes and how those changed as you change the sort of, how well connected the parameter is, And in the spring network kind of situation, the connectivity, how well connected the network is also related to, it's coupled to what is your occupation probability of bonds, right? You can't really decouple them. So those parameters are roughly, I would say, like are proxies for each other. Yes. So when they say adhesion-dependent cell connectivity, they mean that, so in the experiments, the cells are connected to each other by different types of adhesion proteins called ecoadherin. So you can also use that as a measure for, how well connected the system is, right? So in the experiments, in the model, so basically what they're doing is they're taking a cell network and are basically converting it to, using those connections to a spring network, right? So the adhesion-dependent cell connectivity is basically talking about that part of the experiments. And again, if you want to, this is a really nice paper. Sonia Bahar, that some of us know, one of the authors is a good friend of many of ours. So I would recommend taking a look at this paper if you're more generally interested in these kinds of questions of phase transitions in biology. Okay, so I don't know, I just wanted to show this. So this even made into a popular science sort of newsletter. And they said that, well, we have a fluid fish for the first time. I don't know if you say it's for the first time, but this is again related to this morphogenesis in zebrafish embryo that I was talking about, right? Where, you go from a solid-like behavior to a liquid-like kind of situation. And again, this was the figure from that paper that I was showing you, right? So they take these cells and they sort of map out the connections, so you can do a Voronoi tessellation or your favorite kind of tessellation and get a connected graph from this, right? And then build a network of springs using that, right? And then, and we were talking about giant clusters. So see there, this is how in the paper they are, sort of saying that rigid clusters are this green. Rigid clusters mean that these have sort of a rigidity, a finite rigidity. Giant clusters, they are showing in red, right? The second giant clusters are the second largest size and so on, right? Giant clusters here are the sort of really big, well-connected clusters. And then if you plot, something, in this case, they're plotting, the force against deformation, but this would be like our shear modulus or effective, medium spring constant, that as a function of, they're plotting the normalized connectivity, but for us, it was like the bond occupation. You see that, it goes from 0 to non-zero at about 2/3, right? And this also brings me, I wanted to remind you of the Feng Thorpe paper, that much of which we sort of discussed in one of our last discussions. So this is, again, the paper that I had mentioned. And it's a very readable paper. it's not difficult to work through this at all, and it's very instructive. So I will really, and it's not even a long paper, so I will really recommend going through this. And so we did some of this calculation. So this was our spring energy, right? So it was basically some, spring constant, and then, ui minus uj. Ui and uj are the deformation at two nodes, and this is the deformation of the bond, dotted with the unit vector of the bond in the, in the undeformed situation. And then, Pij is the probability whether this bond is there or not, right? And what they found, what Fengthorpe and Garbozzi found, is that there is a percolation transition at this 0.67, right? And then, it increases. And C11 and C44 are two different types of elastic constants. C44 is the shear modulus, so it's often written as C44, right? And C11 is basically, it's elongation, compression, so you can think of it like a bulk modulus kind of thing. But again, this is the percolation transition point, right? And you can see that the previous figure that I showed you from the zebrafish paper and this paper, that this behavior is like the transition point is the same. So it was kind of cool that the zebrafish folks found their transition to be happening at this 2/3, right? Questions, comments? Yes. So in this case, the order parameter is the elastic modulus, the shear modulus. They were measuring the shear viscosity in case of the shear instead of the shear modulus, but the actual thing would be if your material, often in biological systems, is viscoelastic. So you should measure the complex shear modulus if you can, which will have an elastic part and a viscous part.
There is no elastic.
Below the phase transition, there is no elastic modulus, no.
Can you say when you're in a regime below the phase transition,
Yeah.
is there something that you can measure that tells you how far you are from this?
How far you, no, not in theory, right? So in
For example, between spins or something, you will still get like a divergence.
So here you can get some things just above the phase transition. Below the phase transition, I guess if you can, I'm not looking at scaling relationships here where we look at approach to the phase transition. So you can study those kinds of things, but we mainly focus on P minus PC, where P is slightly above PC, not where P is slightly below PC.
You could look at finite size, shear modulus at a finite scale. And so as you go to a larger scale, as you approach closer to transition, that scale where you lose your shear modulus will get larger and larger.
Okay. Oh, yeah. And that made me think of something else. Yes, there is another quantity we often look at. We look at something called a non-affinity parameter. And this non-affinity parameter also will show divergences at the shear modulus. So you can calculate that quantity both below, like you can calculate that everywhere, right? And what is the non-affinity parameter? So basically, if you had a completely ordered connected lattice, right? Then if you apply a shear or whatever your deformation, every node will move in a certain way, in a prescribed way, will follow a rule, right? But then once your lattice or your network is disordered, then your nodes may not follow that deformation exactly. So a non-affinity parameter is basically, it's a measure, it basically first measures the difference between where the node is in the actual disordered network is, minus where it would have been if you had a completely ordered affine network, right? And then it takes the square of that. So that quantity you can measure throughout. And what that quantity will do is it is going to show, so it will have something and then it's going to show a divergence at this point, right? So that's a quantity you can measure. Yeah. And what Leo said is also correct. For finite system sizes, you can also look at. Yeah. The viscous, I'm not talking about the viscous modulus because, I'm really not taking into account any kind of time dependence in the kind of theory I'm doing, right? But for people who study, transiently cross-linked networks where the cross-links can come on and off, there you also look at things as a function of frequency and you can make a comment. You can comment about the viscous modulus. In this limit, we can only comment about the storage modulus, not about the viscous modulus, because there is no dynamics in the system. At the end of my talk, I can bring up the non-affine parameter figure and show you what that looks like. Okay, so this was only spring networks, right? And we are still doing recap, what we already did yesterday a little bit. We also looked at what will happen if you have semi-flexible networks, which is basically in addition to the spring energy, you also have an energy from filament bending, right? So initially filaments, so you can think of a filament as a series of bond pairs, which constitute these hinges. And any hinge which was initially straight like this, but now has bent, what is the cost of bending of that hinge, right? So that's the angle, the theta is basically this change in this angle, right? And square of that, and P, there are two P's here, Pij and Pjk, because, for there to be a contribution to bending energy, both of these bonds have to be present, right? So then you have a deformation energy, another contribution like this. So then what we can do is we can write down this total deformation energy. We can do an effective medium theory calculation like we discussed last class, or we can also write down the deformation energy, apply either shear or compression or whatever your chosen type of external deformation at the boundaries, minimize the system, and then computationally calculate the shear modulus or whatever other modulus at different values of P, right? So, and this is how the two things compare up, right? So if we did not have any bending rigidity, the percolation threshold is at 2/3, which I was talking about. But if we do have bending rigidity, then what happens is the percolation threshold, now we have this extra constraints from bending. So the threshold moves to smaller value of P, and it's now like 0.457, right? And I had showed you how you get this 0.457. Because now, instead of just P equals 2/3, we have P plus P square equals 2/3, right, from effective medium theory. And this, I'm showing you what happens for different values of bending rigidity, right? So when the bending rigidity is very, very small, you can see that there is this kind of inflection here, right? And so basically, if bending was 0, it would have gone through a phase transition. So it starts falling, but then it realizes that there are still additional constraints, right? Bending is actually not 0. So then it, doesn't really fully go through. And then you see this rigidity transition here. So one of the places where people actually study, what's happening in terms of exponents and etc., is the rigidity percolation point, but also this isostatic point. So this is a very interesting point, the 0.67 thing, right? Because you can see the behavior here versus the behavior for the orange and the green versus the red and the magenta is very, very different, right? So the orange and the green, the bending stiffness is really large, right? So if the bending stiffness is really large, or if your network is really like well connected, densely connected, then the mechanics of the network is mostly dominated by stretching of fibers, right? And they will deform in a sort of uniform or affine manner where, if you look at different parts of the network, the deformation is roughly going to be very similar, right? But in this region where you see this is happening, when the network is very sparse or the bending rigidity is very weak, right? In those kinds of situations, this part is now dominated by bending of fibers, right? And here and your deformation is not going to be affine. If I measure the non-affinity parameter, it's going to be much larger in these regions than for those curves, right? So basically, in this case, if you look at different parts of the network, or if you look at different scales, the deformation is going to look quite different. And for these guys, or for this guy, we can sort of use a continuum approximation, to describe what's happening in the network. But here, the continuum approximation is not going to be as faithful to what you actually see. And when I say what you actually see, I mean either if you do simulations, what would you actually see? Or if you are doing experiments where your networks are often quite sparse, what you're going to see in that situation, right? Yes. No, they are different. So so the mean field theory actually describes this critical point quite well, the isostatic critical point, right? But this is a little bit more tricky, right? So they are different critical points. In a spring network, these two guys are on top of each other, right? In a spring network, if there was no bending, the isostatic critical point is also the rigidity percolation threshold, right? But in addition to central forces, if you have, it's not just a spring network. If you have just a central force network, this is the only critical point you have. But in addition to central forces, if you have filament bending or if you have angular crosslinks, then you have the second thing. For very stiff network, basically, the reason you can't see it because stretching energy completely dominates, right? So you don't really see that at all. I guess I have not done an RG analysis, but for these kinds of values, I would be skeptical. But the thing is, even in experiments, let's say experiments are not here or also here. So for actin, the ratio, this non-dimensionalized ratio is of the order of somewhere 10 to the minus 3 to 10 to the minus 4. So it's this cyan and blue, and if it's closer to the cyan, it's very difficult to see the inflection. There might be, but but experiments are also very hard and messy, so you may not exactly see it, even though it's there. But here, I think if you try to calculate the approach, like the exponents as a function of, if you plotted things as a function of P minus PC and stayed very close to PC, you will see some things. Other questions? Okay, and then still doing recap. And then we said that, okay, so if we added these bending energy, our percolation threshold moved from 0.67 to about 0.45, 0.46. On top of that, if we added other constraints that will constrain, angle between crossing fibers, like Leo was suggesting that other day, and you can see this in networks of, actin cross-linked with filament or actin branches growing, with ARP23. There you will have this additional angle constraining crosslinks. And if you did the effective medium theory or minimize the energy, then what you will see is that the percolation threshold moves even further to this side, to the left, to the right side, and it's like 0.36 now. So basically, if you add more and more constraints, then the network, you can have rigidity with less and less material. So you can ask, how much further can this move, right? Can we keep going down all the way to 0 as you add more? That's not true. You stop at connectivity percolation. So connectivity percolation threshold is usually less than rigidity percolation threshold. That's where you stop when you have all possible constraints that you have wanted to add and you've added them. So. And then final recap is Marcus. If you add constraints, it will make it more affine, yeah. So it will make things a little more affine as well. Yeah.
Constraints needed to get rigidity percolation right at connectivity percolation? Is it just like every vertex is like perfectly stiff?
Not perfectly stiff, but you have angular constraints at every vertex. We actually have that in this paper. So we show that where they will actually lie on top of each other, and they do, yeah. Yes.
Just the connectivity percolation transition. What is the thing that you typically measure that says that there's a transition?
So in a connectivity percolation transition, like your friend was earlier saying, you will look at a connected cluster, right? How big is your connected cluster?
Does that translate into something?
So connectivity percolation often, so kinds of systems where connectivity percolation is interesting. So let's say if you look at a network of rivers, right? And if the water is going to flow, or if you look at a network of, electric resistors and see if there will be conductivity, throughout the network. So for those kinds of things, you will use a connectivity percolation. Also for disease networks, right? So, which is not as nice a network to think about, yeah. And then the last recap that I wanted to mention before we move on was this, right? So in these kinds of, semi-flexible networks, now if you introduce correlations in structure, right? So now you have spatial inhomogeneities. So basically for the same value of P, you can change the degree of correlation and you can get, more connected regions and less connected regions instead of, homogeneously distributed regions, right? And what I had showed you in last class, in that case, we don't do an effective medium theory because it's more difficult to write down the distribution of elastic constants, etc. But we can still do an energy minimization and calculate our moduli that way. And what I showed you is that initially, as things are more and more clustered, my percolation threshold keeps moving to smaller and smaller values of P, but then it will not be monotonic. At a point, it will start coming back, right? So this is what we see. And that's because initially, as you have more and more connected, regions, you can have a path of force transmission for, over long distances, right? With fewer material, fewer filaments. However, if you have too much correlation, you can have completely disconnected islands, right? Where you can't have that. So there is basically, a moderate amount of correlation is good, but like everything in life, too much correlation is probably not good. So everything in moderation, right? So there is sort of an optimum window. All right, so this is where we had left off in last class. Yes, question. Yeah, so this is something that, okay, this is something still to be still ongoing work. And we are trying to understand whether this is still, yes, it's still the same universality class. But the thing is, but there is a series of PC, there is a series of exponents also, right? You remember from last lecture, it's not just one exponent. So this part, like looking at if, what's going on with this transition is a work that's still, in progress. Okay, so for mechanics of cells and tissues, to remind you again, both cells as well as tissues are made up of, networks, right? And that's largely responsible for their mechanics, networks of fibers. And these networks are often disordered, right? And we saw that we can use rigidity percolation theory to sort of understand or characterize the mechanical response of these network-like structures. I also showed you that these networks can have spatial inhomogeneities and how things will shift when that is the case. Today, I want to discuss in the time that's left a little bit about materials that are composite or multi-component, and then what happens when you have activity briefly towards the end. So why do I say materials that are composite? So in this case, the cytoskeleton, once again, we have actin, intermediate filaments, and microtubules. And remember, microtubules on the scale of a cell are very stiff, almost like rigid rods, right? Because their persistence length is of the order of millimeters. So on the scale of a cell, let's say 100 or several hundred microns, you will not see much bends or anything in them. Actin, on the other hand, is semi-flexible, and their persistence length and their contour lengths are comparable of the order of 10 microns or so. Intermediate filaments are similar, right? So it's not just a network. The cell is not just made up of network of one type of filament. It's made up of a network of different types of filaments with very different stiffnesses, right? Similarly, for extracellular matrix, you can have very stiff elements like collagen, but then you can also have a softer network made up of hyaluronic acid and proteoglycans and so on, right? So in real cell, so in vitro systems that you can construct from purified extracts, you can have network made up of only one type of filaments. But in live cells and tissues, things are always multi-component, right? So then how do we begin to study these kinds of systems? And I'm going to frame this question by using an example of a tissue that I work on with my collaborators at Cornell, one of whom is actually going to be at the summer school during the last week. So you guys should feel free to ask Itai more questions related to the experiments when he is here in the last week, right? But I want to use this example of a real life tissue to tell you how we do these kinds of, how we use rigidity percolation to characterize mechanics and mechanical phase transitions in actual biological systems. So the tissue that I want to use as an example is articular cartilage. So this is our model system. So articular cartilage is a very remarkable tissue. So it's only a few millimeters thick, so 2 to 3 millimeters thick. It's present in our joints, in our knees and our elbows, right? And it cushions our long bones, right? And it's made up of mainly extracellular matrix. Now, extracellular matrix doesn't make itself, right? It's created by cells. So there are cells called chondrocytes in our cartilage, which make this ECM. So you guys still have a lot of chondrocytes in your cartilage, and you had even more chondrocytes when you were babies. But once you hit your mid-30s and get older, there are fewer and fewer of these chondrocytes left. So then cartilage has very little regenerative capacity once we have crossed over to our 40s and 50s and 60s. However, even after that, people still run marathons or go hiking or even do our normal stuff, running, jumping, walking, etc. So over our entire lifetime, our cartilage tissue undergoes several 100 million loading cycles, right? Anytime we do any of the. So then you can ask, even with this little regenerative capacity, how does it last us, our whole lifetimes? What is sort of the physics underlying the mechanics of this tissue, right? And then when our cartilage does get damaged, either because of, injury, you can get injured while running a marathon or while hiking if you're not careful, right? So if cartilage does get damaged, it would be nice to have sort of good replacements for cartilage tissue, especially for those of us who do not have very many chondrocytes left, right? But the problem is one of the materials that's used as a substitute for cartilage, for cartilage replacement is the same thing that is used in breast implants, artificial breast implants. And you can all imagine that it's not a good replacement for cartilage, right? So one of the reasons for studying cartilage, especially now with all the advances in additive manufacturing and bioprinting, right? It would be nice to have design principles so that maybe we can come up with better cartilage mimics, etc. And also, let's say if somebody gets injured, being able to make predictions whether we need surgical intervention or the tissue is not injured as badly and it will be able to heal itself, right? So all of these things, can be sort of made better with quantitative understanding from physics and what physical mechanisms are governing these kinds of materials, right? So my collaborators on this work are Itai Cohen, who is in the physics department at Cornell and who's going to be here the 4th week, and Larry Bonasser, who is in Cornell tissue engineering department. And the experiments that they have done is mainly from cartilage on cows, but they also have access to cartilage from horses because they collaborate a lot with the veterinary school at Cornell. There have been none of this is on, tissues from humans, but these are all large mammals. So the hope is that, at least some of these things will translate, right? Okay, so what does the cartilage extracellular matrix look like if I just look at a, spherical cow version of cartilageous ECM? So it has this collagen fibers, which are these, stiff, thick guys. And if you want to know more about collagen, we have an expert in the room, Angela. She can tell you, during, after I'm gone, if you want to know more about what collagen is like and, why it has, its structure and properties, I guess you are an expert, but, so it's made up of a network of collagen fibers, but there are also this brush-like, polymers, but then they're connected to this hyaluronic acid chain. So you can think of it in my spherical cow picture as a second flexible network or a flexible sort of elastic background or something, right? But the collagen network is not there just on its own. So now what can happen is as we get older or when we get, injured, etc., we can lose constituents, right, from this tissue because of either mechanical wear and tear or biochemical degradation, right? And the mechanics of the tissue is actually quite depth dependent. Like I told you that the tissue is a few millimeters thick, right? So close to the joint, the density of collagen and aggrecan, all of this filamentous material, that's quite less. It's quite sparse. But as you get close to the bone, the density is quite large, right? So the density of or the concentration of collagen and this aggrecan changes as you go, from the joint part to the bone part, right? So it's depth dependent mechanics. And, if you lose some amount of material, it depends where you're losing it from, whether you're losing it from close to the joint or where you're losing it from close to the bone. In one case, it may not affect things very much, and you will see in which case, and in one case, it could be catastrophic failure, right? And at large strains, the behavior of the tissue can also be highly nonlinear. So, so these all make this a really complex system. The other thing is that if you want to make predictions about how the mechanical function of the tissue is changing during injury or disease, it's really hard you're not going to sort of take a chunk of cartilage from your patient and tell them that, okay, I'm going to take a part of your cartilage and study it and then tell you what's going on. That's not the way to do it, right? But if we have a framework that can map sort of structure and composition to mechanical properties and function, right? Then we can get MRI images and information from those kinds of things, right? And then be able to predict mechanics. So that's sort of, one of the goals of what we are trying to do. Now, before I talk about shear, I want to talk a little bit about compression. So when you are sitting, etc., or your cartilage is most of the time under some compressive stress, right? And the behavior of this kind of tissue under compression is actually well understood. And the theoretical framework that's used to understand that, this goes by the name of poroelasticity. Basically, you have a porous skeleton, in this case of collagen, and you have, water and hyaluronic acid, etc. And how they interact to give you compressive properties can sort of explain cartilage's behavior under compression. So this has been the cornerstone of cartilage's compressive properties for the last, 3 decades. And this is more or less well understood. However, you can imagine that if you think of cartilage injury or fracture or damage, the events that are involved there are mostly shear events, not compressive events, right? And what happens when you shear cartilage is not as well understood. So the way we went about building a framework is we said that, okay, what we are going to do is first measure composition of healthy cartilage, right? So what is the concentration of cartilage? Healthy cartilage. What is the concentration of collagen? What is the concentration of aggrecan? Aggrecan is the largest of the other proteoglycans. And what is the shear modulus, right, for this healthy cartilage? So you can plot the shear modulus as a function of aggrecan concentration and collagen concentration, right? We do this first for healthy cartilage. Then what you can do is you say that, okay, now I'm going to look at a different part of phase space, and I'm going to take this enzyme called trypsin, and I'm going to digest away the aggrecan from a large part of the cartilage tissue. And now I'm going to measure the shear modulus of this thing again, right? So now I'm looking at degraded cartilage. And now, you have degraded away the aggrecan, so there is only collagen left. And this is what the shear modulus looks like, right? So we took different regions in phase space of cartilage, one healthy cartilage, one degraded cartilage. And so we have different concentrations of collagen and collagen and aggrecan, and we have different shear modulus. And this is what it looks like. And then what we say that, okay, now we basically are going to try to build a model which fits these curves for the healthy and the degraded region, and then extrapolate this model to sort of cover other parts of, phase space where we don't have experimental data, which means that if I had a disease pathway that went in this direction or this direction, I will be able to predict what the mechanics would look like. Yes. No. Yeah. I mean, you will have some tissue with aggrecan, no collagen, but it will not have the properties of, it will not have the load-bearing properties of cartilage, because that's mainly coming from the collagen network. So in the interest of time, maybe I can skip some of these things and I can tell you more. This is basically showing you experimentally how they measured concentrations and shear moduli of collagen and aggrecan, etc. But in the interest of time, I'm going to skip to the model. So the model that we use here is very similar to what the model we had written down for semi-flexible networks, right? There is a spring part of energy, and then there is a bending energy. Instead of writing theta minus theta naught, I'm writing it in this form. It's essentially the same thing. Uij is the deformation of this bond, and Ujk is the deformation of this bond. You can ask, why is there a plus sign and not a minus sign? That's because if I had a minus sign, it would have been Uij minus Uik, like basically Uji minus Ujk, something like that. And because I have shifted these guys, I have reverse the I and the J, we end up with the, with a plus sign and not a minus sign. And then, you can see that we have this additional term. So you can ask, what is this additional term? The additional term is because now we don't just have a collagen network, we have a collagen network that's embedded in an elastic background. So if there are transverse deformations of the bonds or the fibers of the collagen network, that's going to be resisted by an elastic elasticity of this background gel, and that gives you this third term, right? And then you can again do either an effective medium theory or a energy minimization. In this case, we did the energy minimization approach, and that's not still done, right? So once we did that, we can get a simulated value of the shear modulus of cartilage. However, that simulated value of shear modulus of cartilage will still go to 0 at the percolation threshold, right? Because all the background gel is doing is adding even more constraints. But the thing is, in the real tissue, because there is some aggregan, and also not all of the collagen is part of the network. There are bits of collagen that sort of break off from the network and become part of the background gel. So that sort of sets a lower bound or threshold for the modulus. So the modulus doesn't completely go to 0, right? So then what we do is we write something of this form. So this is a part, I will show you what things map to. So basically, this guy is coming from this is coming from the simulations, and then we have another elastic sort of background, which is coming from the contribution of things that are not part of the network. And things that are not part of the network, you have a hyaluronic acid contribution that's same throughout the tissue depth. We have a contribution from collagen, which are pieces of collagen that are part of the background gel that can change with height. And then you have aggregan that are part of the background gel that is also going to change with height. One question that nobody asked me so far, so I will ask myself and answer myself. So I'll answer. So I told you that the tissue is 3-dimensional, and the model I'm showing you is a 2-dimensional model. You guys didn't ask me, how the heck are you using a 2D model for a 3D tissue? Okay, so this is what's happening. So the tissue is 3-dimensional, right? But remember, at different depths of the tissue, right? The lowest depth is basically at the joint. That's the mental picture. And then higher, I'm thinking of it's getting close to the bone. So at different depths of the tissue, the concentration of collagen and aggregan is different, right? So what we do is we basically, like if you were doing an experiment, if you are doing confocal microscopy, for example, you will have a series of Z-stacks, right? Which is a series of slices of the tissue, 2D slices of the tissue at different depths. So our mental picture for the theory is the same thing. We have a series of 2D slices of the tissue at different depths. And for these different 2D slices, the density of collagen and aggregan is different. And it's informed by the measured density of collagen and aggregan for that tissue depth for the tissue. So this is how we convert a 3D tissue to a series of 2D slices. What's missing out in this picture is basically you also have, connections and correlations in the Z direction. But if I have this 2D slices, I don't have that, right? So that's something that's not here. Okay. So now with the assumptions in the picture that we do have, what we are able to do is, let's say you can look at, a slice where, you have basically very little collagen, no aggregan, a lot of collagen, no aggregan, a lot of collagen, a lot of aggregan, a little bit of collagen, a lot of aggregan. You can look at all these situations will belong to these different parts of the phase diagram, but you can look at any other kinds of, combination of composition. And you can sort of, expand out. So these solid lines are predictions of the theory when fitted to the experimental data, right? And then these other parts are basically extrapolated versions of this theory. So the next thing you can ask, okay, so what are the things that we know and what are the things that we made phenomenological assumptions? Because that's another big part of sort of when we use a model, construct a model to understand and predict experiments, right? So properties of cartilage, collagen has its properties, right? Its stiffness, its persistence length, aggregans, concentration, hyaluronic acid has its overall property. Hyaluronic acid concentration, by the way, is not changing, right? And then collagen will have its own concentration. Collagen will have cross-linking. Collagen will have connectivity, right? And what are the things that are being in the model, which parameters are being influenced by these properties? So collagen elastic properties in the model will basically inform the stretching and the bending modulus of the stiff fibers, right? Aggregate concentration is basically the background gel modulus part, this part, right? Hyaluronic acid concentration is the background modulus floor, which is not changing as a function of depth. This part is changing as a function of depth because this rho is going to change as a function of depth. Collagen cross-linking and collagen connectivity is going to, and collagen concentration is going to affect both of these guys, right? All of these guys. So this is how sort of the theory parameters are related to parameters from experiments, right? And this is what, again, we used. Okay, so essentially what I've showed you is how we can use a rigidity percolation-based framework to create a model that connects composition to mechanics in a tissue like cartilage, right? And it can predict mechanics during pathways of disease. If I went this way or some other way, it will tell me what the shear modulus should be in that situation, right? And we can combine this with structural information from MRI images and such to sort of, make some informed guesses about patient joint mechanics. And this kind of a framework, I used it for cartilage, but you can have it for any other kinds of system where you have a stiff network like collagen and a softer network like aggregate, etc. Right? Any questions for this part? Yes. Yes. We are not modeling that part. No. So there are other things. There is synovial fluid, there is meniscus, and people model all of those things also. We are basically just modeling not what's happening at the joint, but the cartilage tissue that's there between the joint and the bone. Yes, Marcus. So this GSIM is, we are getting this from the rigidity percolation thing, right? And this will have a rigidity percolation kind of dependence. So it's not a linear something, right? At the percolation threshold, this is going to go to 0, even with all the constraints, right? But in reality, for your tissue, your G is not going to go to 0 at a certain concentration because you are setting a background floor because of the presence of all of these materials. And this part is taking care of all of that. But this part also changes as a function of tissue depth. The hyaluronic acid part doesn't, but the aggregate part and the bits of collagen that are not in the network, that part are both going to change as a function of tissue depth. So yeah, so that is a picture. The next picture I use is that, and I'm going to show you that model. So so the thing is, in science, we don't go from something, or I don't go from something complicated with the get-go. I start with the simplest thing, add the next layer of complexity, add the next layer of complexity, and do it that way, right? So that's what we did here. Here we thought that, okay, it seems to work well, so let's just use this. But then I go to the model that you just described, and that works even better for. And if you want to study things like fracture, then this is not going to work. So there you need to have that secondary network, right? So that's indeed what I wanted to, yes. So the thing is, here I'm looking at everything from an elastic point of view, right? I have no plastic deformation or anything. So there may be hysteresis, right? Because, if you load repeatedly, but this model is not going to capture that. Okay, so now I also want to talk about a little bit about what happens when you have large deformations. For example, how will cracks propagate in cartilage, right? So the way people study crack propagation and fracture in cartilage is they make a small notch in the tissue, right? And then they sort of apply, stretch the tissue like this. And what you see is that this notch or this crack sort of blunts like this in cartilage, and the bottom sort of pulls out like this, right? And I also wanted to show you another interesting thing. I guess I have time. This is not cartilage at all, but another example. And you can see that, this is not, it's not showing brittle fracture, for example, like you will see in glass or a sheet, of something which was, more like rigid, right? This is a completely different thing. It's not cartilage at all. Okay, so I will explain what's happening. So this is a cell which is known as, it's a single-celled organism called Stentor ceruleus. It's found in freshwater ponds. And what is cool about this particular cell is if you wound it, if you make a hole in it, it will close that hole. If you chop it off into multiple parts, so long as each part has a part of the macronucleus, it will make a new stentor. It's very resilient to wounding. Its cytoskeleton is a little bit different from our cytoskeleton, right? So, but again, it's very fracture resistant, etc. So for these kinds of materials, both in the context of tissues like cartilage and cells like stentor, why are many of these materials, so resistant to fracture? Why are they so resilient to damage? And one of the reasons, yes. Not bone. I'm talking about cartilage. You're talking about bone?
Well, I think that presumably, I guess you have fibers pointing in a given direction, right? Most of them pointing in one direction. So is this track propagating orthogonal to those fibers, along the fibers, or something?
I mean, the fibers in this case, so you are thinking of the fibers are aligned. The fibers need not be aligned because, again, the tissue has fibers, it's in all mostly. If you're looking at really large strains, you will begin to see some alignment, but you start out by things which are sort of, isotropic and homogeneous, right? So, yeah. But I will show you how we model it. So we thought the simplest way to model it is the next iteration that Marcus's question sort of, helped, sort of think of, maybe you guys already start visualizing. So instead of having a background gel, we say that we have a collagen network, which is now interacting or is coupled to an aggregate network, which is a second network. But the second network is now a flexible network, right? So there is no energy cost of bending the fibers in the second network. And then these two networks are sort of interacting with each other at the midpoints of bonds of the corresponding networks, right? So not at the nodes, but the bonds are sort of connected to each other by weak Hookean springs. Only when bonds in both of the networks are there, right? So this is sort of the simplest picture, mental picture, or cow model picture of a composite network, and this is essentially a double network. Now, I could have also had this flexible network be, semi-flexible or stiff, but in this case, for cartilage-specific things, it's aggregate hyaluronic acid secondary network is more flexible, so we drop the bending part, right? So before we do anything else with this network, we wanted to look at the percolation threshold and the shear modulus, etc. How does that look like, right? So once again, we only apply a small shear deformation, and then we look at the normalized shear modulus. Normalized means that it's normalized by the value where the network is completely ordered, where all the bonds are present, right? And along the x-axis, I'm varying the occupation probability of bonds in the stiff network. Along the y-axis, I'm varying the occupation probability of bonds in the, not y-axis, for the legends, occupation probability of bonds in the flexible network. So in the case where we had only one network, right, and I'm using a Kagome lattice-based network, by the way, not a triangular lattice, right? For reasons that I had described previously, why Kagome lattice. So if there was no secondary network, the percolation threshold would have been here. But as you have a secondary network and more material in the secondary network, you can see that the percolation threshold shifts further and further, right? So you need less material in the stiff network if you have constraints from the other network, right? So this essentially just means that. The normalized shear modulus of the secondary network is also, of the composite network is also larger if you have more and more material in the secondary network, right? Also from the constraint stuff. Now you can ask, why have I drawn two dashed lines? That's for the following reason. So if I was in a region of cartilage, which is very close to the bone, right? My collagen network and aggregate network are both super duper dense, right? So there, if I sort of decrease, let's say I keep the collagen density fixed here and I decrease the amount of aggregate, the change in shear modulus is very little, right? It's not much. But if you're close to the joint, then the collagen network is above its percolation threshold, but it's not that dense. And now if you change the amount of aggregate, you can see that there can be several orders of magnitude drop in the stiffness. So if you lose aggregate from your cartilage, it matters whether you're losing it from close to the joint or close to the bone, right? And close to the bone, it's also for physiological reason our bodies want to have more dense thing because you want to protect the bone from damage, right? So, okay, so this is still with, but you can see how the percolation threshold is tunable, because of, and the tissue can be close to a phase transition just based on how much collagen it has, but then by adding different amounts of aggregate, you can move away from that or closer to that, etc. So then you can ask what will happen if I had much, now if I look at large deformations, right? No longer just small deformations. So now we also create a notch to look at fracture. So we create a notch like this. We apply uniaxial strain at the two boundaries, so no longer shear. And we start with 1%, and then we go all the way up to 70%, right? So really large deformations. And we make this assumption that for the stiff network, bonds are going to buckle, right? If they're compressed to 95% of their original length or more, and they will break if they have been stretched to 120% of their original length. And when this happens, we basically just remove that bond from the network. It's not going to contribute anymore, right? For the flexible network, bonds are not going to buckle, but they can break when they have been stretched to more than 200% or twice their original length. And once again, we remove the bonds, once they have crossed this threshold. The other thing that we say is that breaking of bonds is an irreversible change, right? Bonds that are once gone are gone from the network. But for buckled bonds, if you remove the compression, right, these bonds can come back into circulation. So that's another approximation we are making. So these are all the assumptions that we have now added to this model to look at fracture. Okay, so we look at two different cases. One case when the collagen network is far above the rigidity threshold. So this is the situation when you're close to the bone, right? And we are looking at the normalized Young's modulus as a function of the applied strain. So initially, as you increase the strain, right, you will see a strain stiffening a little bit, right? But then what happens is it reaches a point where your bonds are now starting to break, etc. And so then you see a softening, right? And for a collagen networks that's super dense, these curves for different values of aggregate in the second network, they kind of lie on top of each other. There is not much change. There is some change, but not too much. But look at this. This is the situation when you're close to the bone and where the collagen network is just above its percolation threshold, right? So it's 6.62. The percolation threshold for this Kagome network situation with bending is 6.6. So this is just above the percolation threshold. And now as you increase the amount of aggregate, you can see that the change in the stiffness, right, is several orders of magnitude. And once again, you will see first some stiffening, then it will reach a peak, and then it will start softening, right? Because when you have strained too much, bonds will start to break and buckle, etc. Yes. Why does it get more stiff originally? That's because in a network, etc., bonds might lie, initially they are sort of in a situation. So I guess there are two ways stiffening can happen. First is that when you're stretching your bonds just stretching your bonds across the, their backbones, right? And their overall stretching energy, right, or deformation energy is increasing. So there is that. Sometimes your bonds can also be in a situation that are not stretched out, right, in a given direction in which you're stretching. So initially, when you're starting to stretch, you're stretching everything along this direction, right? And you're also sort of starting to align your filaments along that direction. And that can also give rise to some additional stretching. So it could be a combination. In a network, it could be a combination of all of those reasons. Marcus, and what's your name?
Anna.
Anna, yeah. Anna, you go first and then Marcus, maybe.
Yeah, it looks like as you add more aggregate, it fractures earlier.
This is an important point, and we will show. I will talk about this. This is very important. Marcus. Yeah. Yeah, so this is very dense collagen network. This is close to the joint where correct, yeah. This is close to the bone. This is close to the bone where collagen is very dense. this is close to the joint where collagen is quite sparse. So basically the point is here, if we remove aggrecan, the change we will see is little. Here, if you remove aggrecan, the change you will see is a lot, and you can have, a lot of damage to your tissue. So which is why we have to be careful about, we also have to be careful about our bones, but there are a lot of joint injuries, and this is one of the reasons, right? Because it's quite thin. Yes. Yes. Yeah. So in experiments, if you do cyclic loading, you might keep breaking bonds, and you might see, like your curves will not completely retrace. In the simulations, I will have to assume, so my PhD student, she also looked at the situation where your starting point is a point where you have broken some bonds and now you have less things. I'm not showing that here though. So this is the thing that Anna was asking, where, that part would be, can I take this question after I've described this figure? So now we are looking at stress versus strain. Once again, this is a situation where the collagen is very dense, right? And you can see that the maximum stress that the network can bear initially increases, reaches a peak, but now bonds have started breaking, etc. So then it falls like this. And when the collagen network is very dense, these curves are all on top of each other too. There's not that much difference. But when the collagen network is just above the rigidity threshold, so still rigid, but it's quite sparse, you can see that as you change the amount of aggregan, two things shift, right? The maximum amount of stress that the network can bear, which is known as the peak stress, that changes. And the more aggregan you add, the more peak stress you will have. However, the strain at which you reach the maximum stress, which is a measure of the extensibility of the material, so it's the strain at which the material will start breaking and fracturing, that strain actually decreases, right? That decreases as you add more and more material. So why is this? So this is a very important point. So as you add more and more aggregate to the network, you're reinforcing the network more and more. So it makes sense that the maximum stress or the peak stress is going to increase as you add more and more material to the aggregate. But the extensibility decreases. So there, before we get to why this decreases, first of all, why is a double network better than just a single network? The reason a double network is better than a single network is, in a single network, let's say if you accumulate stress in localized regions, the only way to relax or release that stress would be by breaking or buckling of bonds, right? Especially if the network was dense. But, because the stress is quite concentrated. In a double network, the second network sort of helps to redistribute the stress over larger areas, right? And if the second network is not very dense, it can undergo low energy or non-affine deformations and movements. It can undergo movements that cost much less energy. But if your second network is quite dense, then you are suppressing these non-affine deformations. And then once again, you're bringing the network to a situation where, there comes a point where it can only release or relax this accumulated stresses by breaking or buckling of bonds. So if you add too much material to the second network, you are suppressing this non-affine deformations, and it will start breaking at lower strains. So once again, too much of anything is not a good thing. There is an optimum window, right? So you want to have a second double network, and you want to add more aggregate so that, you have the desired load bearing, etc. But maybe do not add so much aggregate that you make the network, brittle and hard, right? So I wanted to show you a little bit of what the fracture propagation or crack propagation looks like. This is when the stiff network is always where, like it's close to the bone, it's very dense, right? So now, and this is the applied strain, this is the amount of flexible material, occupation probability. You can see that this is more or less always brittle, right, in this case. But here we have this window where, you don't see brittle fracture, right? It sort of looks more like what you would see in cartilage, where the crack sort of blunts, right? And the bottom is pulling out. And this sort of falls apart at strains of like, this is the strain. So it starts falling apart at strains of, let's say, 55% or so, but it's still tenuously connected. But here, things had broken for the same situation, things are broken here, right? So you can see that. Questions? Yes. Yeah. So basically the overall rigidity percolation of the overall material, because these are two interconnected networks. So the overall threshold of whether the material has a finite shear modulus or not will depend on P1 and P2 both, right? So the way I was showing you things is that I was showing you this as a function of P1, but for different P2. But I could have also drawn a surface curve, but it will depend on both. Yeah. Yeah. Oh, no. When I say close to the rigidity threshold, I mean it's close to the rigidity threshold for the bare network. Thank you for asking for the clarification. Yes. So for the bare network, the percolation threshold is at 60%, and 62% is just above the percolation threshold for the bare network. So when I say far above and just above, it's for the bare network situations. Thank you for asking that. Yes. Yes. Okay, so this is a phenomenon. So for collagen, breaking the collagen bonds at 120% and buckling below 95%, those are informed by what happens for collagen fibers, and there is something known as an ultimate tensile threshold for materials. An ultimate tensile threshold is basically the limit at which they will break under tensile strain. And for collagen, we have some kind of a knowledge of that. For aggregate, we don't really know. So we're like, okay, we know for aggregate, it has got to be much large. What is a reasonable number? We're like, and then my experimental collaborators were like, maybe 200% is like a reasonable number. So for aggregate, it's phenomenological. For collagen, it's also somewhat phenomenological, but it's more grounded in reality. Yes. All right. Okay, so a little bit more for these kinds of networks. We can also use these kinds of double networks to describe cytoskeletal networks like actin and networks of actin and microtubules, right? So in that case, both of the networks will have both stretching and bending energy. However, actin can also have, myosin motors, etc., right? So the way that, we and other people incorporate the presence of motors in these networks is we say that the rest length of the bonds in the actin network when motors are there. So we say that a fraction of bonds are going to be contracted because of the presence of motors. And the way we implement this contraction is by saying that every time there's a motor associated with a bond or a series of bonds in an actin network, the rest length is going to be shorter than the, equilibrium length, right? Than the, what it otherwise would be. So by doing this, and then we can write down energy. Deformation energy has very similar form. We can sort of generate networks with different concentrations. So here, phi A means the area fraction of actin versus microtubule. So 0.25 means actin is 25%, microtubules is 75%. And then, 0.5 means actin is 50%, microtubules is 50%. And then Cm is the concentration of myosin, right? So, what concentration of this contractile fibers you have added. And then you can sort of generate this library of networks for different concentrations of actin and microtubules and motors. You can measure, you can make predictions about what the shear modulus would be. And then I'm not showing you any of the experimental data from this paper, but this paper again had both experiments. So now this is not cartilage, this has actin, microtubules, and motors, but it's again a paper which has both experiments and some predictions from model. So you can use these kinds of models for also cytoskeletal networks. Did I see a raised hand? Yes. Yeah. So ideally, we should do that. That's, again, thank you for asking that question. Ideally, we should do that. We have not taken that into account here, right? The persistence, well, we have not taken into account that in the mesh size. What you are saying is the mesh size should be different. here the mesh size of both the networks are, when mapped to experiments are of the order of like 200 to 500 nanometers in that scale. But what we did do is the ratio of the elastic constants like kappa 1, alpha 1, kappa 2, alpha 2, those ratios reflect the ratio of stretching and bending in actin, stretching and bending in microtubules versus bending in actin and bending in microtubules, right? Because bending in microtubules is 3 orders of magnitude larger than in an actin, or stiffness, not bending itself. So far, I've only shown you things on lattice-based networks. One of you had asked me, what about networks that are not lattice-based, that are off-lattice networks, right? So people also study these kinds of systems on off-lattice networks. In some sense, they actually mimic biological networks better. But the reason I do things often on a lattice is because then I can also make some analytical calculations and predictions that I can compare my simulations with. But this is an example of what an off-lattice network would look like. And again, it has, this case just has actin and myosin motors, and myosin is modeled here as 4 dipoles, right? And it's from a paper by Vivek Shenoy's group and Soft Matter 2010. And what they found, so they found some interesting stuff, which you can expect, again, makes sense. If your network is very stiff already, right? Our network is not very stiff, but the cross-linkers are very stiff. So they don't allow reorganization or rearrangement of fibers very much. Then as you add myosin, not much changes, right? So this is basically showing, the black one is no myosin and the green one is with myosin in the case where the network is very stiffly cross-linked. So this is a very stiff network. This is sort of somewhat analogous or a cousin of the stiff network from my cartilage thing, right? But if you have a very flexible network or a network cross-linked with flexible fibers, then as you add more and more myosin, you can see more and more stiffening again, right? So the goal is basically showing you that if you start with a network which can propagate rigidity, but is not super rigid, it's somewhat flexible, you can modulate its properties much more by adding other things, whether it's motors or it's a secondary network and so on, right? But I also wanted to show you this graph, this result, because it's showing you things from an off-lattice network, which is also something. So the model, this model, like having things like sticks. So here, what they do is they basically throw down a bunch of sticks in a sort of random way, and every time 2 sticks connect, they say that there is a crosslinker, right? This kind of a model is known as a Mikado model. All right. Questions? Yes.
And then you had already a very dense network that make more simulations.
So I think I can map this not to that situation, but I showed you just a version where I had a double network with motors. Instead of having a double network with motors, if I had a single network with motors, it should be able to recapitulate these things if I am able to. But in my case, I cannot separately have crosslinkers. So what I will have, the 2 things will be different, is one network will be very rigid because P is very large, and one network will not be as rigid. It will be percolating, but P is smaller. And then in one network, as I vary the concentration of motors or these contractile parts, things should not change too much. And in the other case, they should change quite a bit. This is not something I or anybody has checked, but it should translate. I guess I want to then, I have some conclusions for you. Basically, that rigidity percolation framework can sort of explain the origin of, cartilage's mechanical properties and predict disease pathways. This paper was published in Science Advances a year and a half ago. And then when combined with a double network model, it can explain fracture properties. This is also a somewhat recent paper, Soft Matter 22. And then double network motors with motors can model sort of actin-myosin microtubule networks. Now, if you want to make materials that are tunable and resilient, right, make artificial materials, so what can you take home from looking at these kinds of things, right? So basically, one of the things that we have learned is that if you take a stiff network, but your stiff network is not too dense, right? It's stiff, or, it's well connected enough that, it's above the rigidity threshold for a single network, then by adding the, appropriate amount of flexible network material or motors or something, you can tune the properties of your material to, however much load bearing capability or extensibility, etc. you want. The other thing we saw that for enhanced fracture resistance, we should try to distribute damage, and we have to be able to allow some non-affine deformations, right? Otherwise, things will become brittle. And I guess with that, I want to thank, so this is my group as of last fall. Two of my PhD students, Lauren and Pansy, have now moved on. Pansy is in SUNY Buffalo. Lauren is working at Pfizer. Rest of my group is still here, and then more people have been added to the group since. And my collaborators, all the names are in blue, are people whose, the figures I mentioned are with papers with, with them, or work done with them. Okay, I do have some concluding remarks. I guess so. I wanted to talk about this a little. So first, I'm going to read it. You can read it. I will also read it. So this is from a paper by Melanie Steffen, and it was published in Nature in 2010. It was like an editorial article. So a couple of months ago, I received a letter informing me that my fellowship application had failed. On the same day, Brazil's World Cup squad announced that the football phenomenon, Ronaldinho, had not been selected. Cool, I thought. I'm like Ronaldinho. But that thought offered only little consolation. No scientist enjoys such failures, but too often we hide them. We don't share it with anybody else, right, when we fail. As scientists, we construct a narrative of success that renders our setbacks invisible both to ourselves and to others. Often, other scientists' careers seem to be a constant, streamlined series of triumphs, especially if you're following their Twitter, right? Therefore, whenever we experience an individual failure, we feel alone and dejected. Such is not the case with every profession. Consider Ronaldinho. A football player cannot hide his setbacks. Everything is out in the open. Every failure is to be selected for a big competition. Every injury, every missed penalty is on display. Maybe this is a good thing. It shows young aspiring players what it means to be a football player. It helps them to cope with their own setbacks. So I wanted to say this because when you read a paper or a series of papers or see the CV of your mentor, you see all of the success stories, right? But science is very non-linear. A lot of hard work goes into things. For every paper that's published, every calculation that lots of calculations, lots and lots of experiments that didn't go anywhere, but still gave you some insights, right? And it's a lot of hard work, but we don't see all of the hard work. We only see the success. But there is a lot of hard work. There is a lot of perspiration. There is a lot of failure. So all of this is very, very common. So the reason I wanted to read this, I feel like it would have, and the author also feels it would be nicer if our careers were also like the careers of, tennis players or football players, where you can see everything, right? For us, you only hear the success stories. But, in a science career, there will be failures, there will be successes. You should not feel alone when things don't work, right? Because it happens to all of us, even though you may not see it. So with that, I would end and take any questions you have. Yes, Leo.
I'm just curious, what was this a part of?
This was not a science article. It was basically the author was suggesting that scientists should make a CV of failures so that their mentees can see all of the situations where the mentors tried hard and didn't succeed. So the mentees will not feel frustrated and dejected when their calculations didn't work or their experiments didn't work. I really like this article. So, Yes, Patricia. Yeah.
Find a way to produce materials which are very resistant to stress, and by doing a double network,
Yeah. Yeah.
And so this strategy works for polymers, but
Yeah.
doesn't seem strategy which is used.
I guess I would say nature also uses this strategy a lot. There is a difference in what's happening in nature versus what's happening in synthetic gels, like JP Gong and other people have been working on it for a while. Is for biopolymers, there are a couple of different things, right? We have, we have a combination of networks that could be flexible, but also could be semi-flexible and stiff. Most synthetic polymers are flexible polymers. But for biopolymers, again, we also have, which something I neglected here, different types of cross-linkers, which can also sort of modulate things and give us a much more rich library from where to design things than in synthetic things. But one of the things I and my friends my collaborators are interested in is making biotic, abiotic hybrid materials, where we will have some of the functionality of biomaterials, but some of the longevity of synthetic materials, because synthetic materials will not denature as easily often as biopolymers, etc. But yes, this is a very good point. Like, there has been a lot of work in synthetic materials, and JP Gong is sort of she's the leader of the field, if you look up her name. And double networks really can be, very, very useful. And, you can have really very large, Young's modulus, and very, that than you will get from just materials made up of one type of filaments, etc.
The myosin network.
Yeah.
In fact, it could be because of the filament, they are dynamic.
Yeah, because of the, yeah. And one of the other things, I also didn't show it in this thing. So we also wrote a paper where we are basically coupling polymerization and depolymerization of actin, which is modeled using, I'm forgetting. So the dynamics of that part is then coupled to rigidity percolation to sort of give you. So the dynamics is modeled through master equations, which can give you how actin filaments are growing or depolymerizing. And then you can connect it to a percolation theory to sort of get the mechanics of the network. But even without polymerization, depolymerization, the network can reorganize, because the crosslinkers can sort of may not be permanent crosslinkers, can be, can have a finite lifetime, can be transient, etc. There are also different types of crosslinkers, right? Because when cells crawl or move, they're constantly remodeling themselves. And they're able to do that because of all of these reasons. So absolutely. Yes. It's not the case. Oh, in cartilage tissue. So in cartilage tissue, it's not the case. No, the cross-linking is kind of permanent. Yeah. Yeah, which is, if that was the case for cartilage, I think we would be, we would be less, so I used to hike without trekking poles until I started doing research on cartilage. And now I always hike with trekking poles. I'm like, I don't want to, yeah. Yes, there was a question. Yeah. Can you speak a little bit more loudly? In an effective medium theory. So I guess the only way to do that, it would be hard, right? Because it's up for a passive network. For a dynamic network, I guess you can do in quantum mechanics, people do a dynamic mean field theory. I guess you can think of a dynamic version of mean field theory for these things, but then that will have to evolve as a function of time, right? So that will change with time. But no dynamic mean field theory exists for this. So if you do it, you will be the first person to do it. Yes.
The networks that show that build up components that have very remarkably long persistence lengths.
Yeah.
Basically, any artificial polymer that I know has like tiny persistence lengths.
Yeah.
Be also the materials properties or the chemistry properties make things that are so steady.
Yeah.
What's happening on the artificial cartilage front to solve that side?
I don't know. So artificial cartilage, the things that are happening on the artificial cartilage front is people are trying to see if you can use stem cells to make, then become chondrocytes and then generate cartilage. But the cartilage has to be, the architecture also has to be right? So the cells still have to be chondrocyte, but the architecture has to be right. So we are more focusing on the architecture part. But some of the other things that I didn't talk about here, because it was not exactly this, is for some of my synthetic biology research, we are combining biopolymers from cells like actin and microtubules with circadian rhythm proteins from cyanobacteria, so that and which will act as cross-linkers if you coat them with biotin and coat your substrate with also biotin. So you can go between cross-linked network to unlinked anything on this 24-hour cycle of the cyanobacteria clock, etc. So anyway, yeah. Other questions? Everybody is hungry for lunch. All right, thank you. Thank you so much. And I